Good. Okay.
So we are recording. Erica, you got a favorite dance move? So my youngest daughter just turned 4, and she did this dance move that totally cracked me up. So now she just does it randomly for no reason. And it's and that is now forever on YouTube. Dr. Og doing, would you call that the dancing, Running Man or something like that?
Was that video?
It's on. Yep. And I can totally see that'll be like the meme that'll just be, it'll be the next meme picture that goes across the internet. I hope not. All right. What is UNC22? Do you know what UNC stands for? Does anyone know it? Uncoordinated. Uncoordinated. Okay. Introduce yourselves.
Okay, I'm Catherine Wright. I'm Corinne.
I'm Erica Blair.
Okay, so we have the first section of this paper. So the researchers were looking into how they were going to determine the effectiveness of RNA interference and whether like sense or antisense or the double-stranded would be more effective. So they first just decided to look at this one gene, UNC22, which codes for non-essential microfilament proteins and the little worms over there that are pictured. And there are several thousand copies of that gene's mRNA present in each striated muscle cell of the worm. So it was pretty good.
So what is a microfilament?
Microfilament is...
Cell biology people?
Microfilament is like in your protein and myosin walks on it?
So yeah, that's right.
Okay. Myosin walks on it and that's how your muscles contract and relax.
Right.
And since this isn't like the necessary-ish one, there's like active microfilaments, but this codes for like a different kind of one.
Great. Thank you. I love it.
So when they tested the UNC22 gene, they were able to describe a semi-quantitative relationship between the dosing that they gave and the phenotypes of the worms. And so in this case, because the interference interferes with creating a protein that allows their muscles to contract, they become twitchy. And so that's the phenotype they're allowed to see. And when they increase the dosing of the double-stranded RNA, then it kind of increased the incidence of the twitchy phenotype. And so quantitative analysis would be able to describe that with numbers, qualitative would be with observations, and there's a semi-quantitative, so somewhere in between there. They don't have a model yet for to describe the relationship between the dosing and the phenotype.
So the assay, to redefine this word assay, the assay is whether the worm can glide smoothly through the media. And what they do is they go around like this, and then they go backwards like this. That's how they move through the environment. If they're twitchy, they can literally be spastic kind of twitchy, like that. But in the case of UNC22, they just look like they're like totally drunk or totally high and just kind of like, hey dude, a little bit, what's going on around here? And they're really, really slow. So it's hard to quantitate that, but you can say that worm is not normal and that worm is normal. And so they count it, and it's a lot of subjectivity to it. Just to clarify more detail, but you did great.
So with their initial findings, they found that some inactivity of UNC22, there was the like slight twitchiness and then not really stellar movement. But when there was complete function loss, there was obvious deformities, and then there's just like no motility at all. So you can, like our meme says, you can have some and still work, but if you don't have any, it sucks for you. And then we have another meme.
Very good.
Bringing it back to frozen. And And that is the first paragraph.
Thank you. Questions? That was great. Questions? If you guys are lucky, I use my Olaf voice, and I'll read that meeting.
That's what I was hoping for.
Group 2, introduce yourselves.
Hi,
Hi, I'm Alex.
I'm Andrew.
Did you email it to yourself by chance?
No, back there.
You emailed it
Is that in here?
It's on here.
Will that fit in here?
There's a
small
fun
right
there.
side
Plugs on
on
the
it.
Oh, yeah.
Does anyone else have it on flash drive email to yourself or? Okay. Wow, we might get through all of them today. Anyone worried? Quick group meeting. Okay, introductions. Oh, you did it already?
I'm Michael.
I'm Andrew.
I'm Alex.
Are you guys brothers? You look a lot alike.
Only sometimes, depends on the day. Okay, so we were assigned the second section of the report. So just in a little bit, we're going to talk, do a little bit of a background. So first of all, the UNT22 gene, it encodes this non-essential biofilament proteins. So kind of like the other group discussed, wild type is smooth gliding and is effective, can be different levels of twitching. And this is actually the gene here. In the paper they have this figure here, and we were actually, our paragraph talks mostly about this A section where they have these strings of RNA that interfere, and so they have these different strings that they use here, and A is the one that, the region that was discussed in our paragraph. So this right here is actually drawing the sarcomere. It's not very good. So sarcomere is the smallest unit of contraction in a muscle. And so here's the myosin filaments, and here's the actin filaments here. And so it contracts and that. So the UNT22 gene codes for a protein that acts within this region here. It kind of associates with the myosin. So also in our, if you go to the next slide, please. There's also this kind of summary table, talks about all the different experiments that they ran. And it talks about our gene is way up at the top here, that UNT22A. And so they tested out sense and antisense, and then a mixture of sense and antisense with different results that we'll talk about in just a minute.
All right, so we wanted to break it down by sentence and kind of explain what's going on. So this first sentence states that basically they had antisense and sense RNAs covering a 472 nucleotide segment of the UNC22, and they had basically didn't have very much success or any twitching unless they had a very high dosage. And so that's kind of what this translation is saying here, that it required an extremely high dosage of only antisense or sense RNA to be injected in order to have any interference with the function of the gene. And then in contrast, which I like to use that as a word, just to be like, okay, so we just said something and said, okay, this didn't work. In contrast, now you kind of expect that this is going to be something that's going to be a different result than what we just had. They had a sense-antisense mixture, and with that, they were able to get interference of the UNT22 gene, specifically at least 2 orders of magnitude more effective. And here's just a kind of recap of the table that we're referring to again. The results are on that F1 phenotype column there, the wild type responses for both the sense and antisense. And then at the mixture of sense and antisense, you have 100% strong twitches.
Okay, so this sentence was a little bit more difficult to understand, but it basically said that
the lowest, they found a lowest dose that would affect a response in this interference. And that was about 60,000 molecules of each of the strands, so both the sense and antisense. And that was the lowest that led to twitching in any of the progeny. So dilution does matter, and they actually found the limit of that. So the next sentence is essentially saying the same thing, but this is demonstrating how they found that out in their actual experiment. So what they did was they injected this sense-antisense mixture into the embryo, and if they injected it too early, the mixture would be too diluted by the time the worm grew up to be an adult worm, I guess it would be. But if they injected it after about 500 cells had formed, then it would be good enough to affect interference. So in conclusion, the sense and antisense mixture was found to be at least 2 orders of magnitude better than just the sense or antisense alone. So it begs the question, what's happening to do this? Because you'd think if it was just the 2-fold increase of either of the strands, just having an increase in the concentration, then it would just give it a 2-fold response. But it's by orders of magnitude greater that it has the interference response. So we had the question, what could be happening, and we didn't come to an answer of that. Is that the end?
Yeah.
Okay. That's the end.
Yep.
Any questions?
That's the sarcomere. That's the sarcomere. The sarcomere. We didn't find a meme in there.
Okay. I'm gonna change. Can you go back a couple slides?
Yeah.
One more. This is it. All right. So, because I understand you guys were looking at one paragraph, so you weren't looking at some of the information that I just presented in the first one. Let me bring these two together, and we'll come to some really important conclusions. Group 1, on their slide, they said there's a couple thousand messenger RNAs per cell of the organism. And what they're saying is, when you inject an embryo with 60,000 copies, you've only got 60,000 copies you've put into the whole organism, and that embryo divides, those cells divide. By the time the organism is an adult, if you've got 1,000 cells, you've only got a few copies per cell, right? And you still get twitching. So what they're basically doing is setting up a question for later on is, how do I have 2,000 copies of the RNA and only 3 copies of the double-stranded RNA that I put in there and still get the twitching effect? Does that tell you that this is binding and inhibiting this, or does it tell you it's got to be enzymatic?
It has to be enzymatic.
Yeah. So that's kind of, this paragraph is a setup for what some other people are going to be talking about. Does that make sense?
Yeah.
That's cool. So you did great. Thank you. All right. Group 3. How many groups are there? 10. 10? I'm hoping group 5 got prepared for today. Did group 6 get prepared for today? Or are we waiting?
We're 6, yeah, we had our stuff ready.
Okay. So we may get as far as 6.
Okay.
Introductions?
Yeah, so we're going to talk about how double-stranded DNA can interfere with gene expression. And we're talking about a paper that got a Nobel Peace Prize. And we're talking about paragraph 3 of that paper. So my name is Nikki Sinclair.
I'm Hannah Bloom.
And I'm Shayna.
Alright, so just to review some terminology, mRNA sense transcript is what codes for the protein. the mRNA antisense transcript does not code for the protein. That's just a really basic review. And so according to our paragraph, the hypothesis that they tested was they were wondering whether or not the sense and the antisense were binding within the cell to create a double-stranded piece of DNA. And so some technology that they utilized to test this hypothesis was gel, which we've been doing. And so they mixed the two, the sense and antisense, and they ran it on a gel to see if it did in fact create the double-stranded RNA. And they found that it did because it didn't run as long as a single strand would have run. And then they gel purified that and injected it into the cell, and it did in fact interfere with protein expression. So this double-stranded DNA interfered.
Double-stranded RNA.
RNA.
Oh no. Oh no. Where did you get that?
Google? It's like the orange picture on Google.
Nice.
Okay, so, like Hannah was talking about that, it was purified double-stranded RNA was already already annealed and it was injected into the cell then so that it would interfere.
I'm blushing. I'm blushing in front of the class before that. Wow. Gold star award. All right, continue.
Oh gosh. Okay, so, but it's not necessary for the double-stranded, for the RNA to be double-stranded before it's injected into the cell. So there's a couple sentences that talk about how if it's, if sense and antisense strands are injected separately into the cell, that it still causes interference. So there's two ways that it talks about that this can happen. One, like you take, you make your mixture in a low salt concentration so that prohibits the formation of that double-stranded structure. Or you inject them sequentially, like one right after another. And in both of these, you still get the interference.
So as Shana was talking about, you can add the sense and antisense or RNA in sequentially. So if you inject the sense in and then you wait a duration that's lower than an hour, you're still going to get the interference and the twitching in the worms. But if you wait for a period that's longer than an hour, you don't see the twitching or you see minimal interference. And this can be due to the RNA being degraded, or it just becomes inaccessible. So if you put the sense in, it degrades or becomes inaccessible so that the antisense cannot find that strand and make the double-bonded or double-stranded structure.
Oh, you did. 3 goals. Very good. I love it.
Alright, see ya.
Wow. Good. Okay, great. So I just want to, I like drawing data. So Hannah was talking about, basically you run a gel. So if you take a tube, and you've got sense and you got antisense, you mix them in a tube and you run them out, the sense and the antisense that will anneal will run at some size, right? If you've got sense that hasn't annealed yet to antisense, it's not as big, and so you'll get a separate band. And you'll probably get another band where the antisense is, right? So probably what they did, and let's just say we ran a whole bunch of this out, so that's a big band like that, and that's a big band like that. So what they did is they said, if I cut this out of the gel and I purify that, so I know it's double-stranded and I inject that in, it works. And if I take this and inject it, it doesn't work. If I take this and inject it, it doesn't work. Then the experiment they said is if I take this and inject it in, and then moments later inject this in, probably these guys anneal in the cell, and you get interference. But if I inject this and I wait, was it an hour, 4 hours, or something?
Anything greater than 1 hour.
Anything great, if I put this in and wait an hour before I inject this in, it doesn't work. You don't get the interference. And the idea is maybe that gets degraded. I don't know, I'm just repeating what you said, sorry. All right, any questions for them? Did they get their applause yet? All right, thank you very much. Group 4, we're doing great.
Alright, so we did paragraph 4. I'm Travis.
I'm Jim.
I'm Mackenzie.
This is what our paragraph looked like. What's that?
Beauty.
Alright, so this paragraph questions basically where and how specifically double-stranded RNA interacts in the cell. And so it's known that a cell has a protein kinase that triggers the panic response only when double-stranded RNA is present. And what they originally thought in the paper was that this panic response was initiated by the interactions of both the antisense and sense strands, and that this essentially interfered with the strand's ability to code for the mRNA, which led to silencing.
So when they did the test, it was they took random genes, RNA, like completely unrelated to UNC22. They just took the dsRNA and injected that into the cell to see if that would activate the mechanism, and nothing happened. And they tested single-strand RNA, single-strand, double-strand of the UNC22 RNA. Absolutely nothing happened with any of it.
And so they concluded that only dsRNA sequences located near the translation site affected the silencing of the mRNA. And the tests were designed so that they could see if it was the panic response that was prohibiting the mRNA from coding for proteins, or if it was the dsRNA. That's basically it. Any questions? All right.
Okay? Okay, one thing you missed on this one, we'll get to that in a second. You guys can grab a seat though. Okay, so this panic response, you can go ahead.
Well, I think the easy way. Yeah,
Okay.
that's good. Yeah.
Okay, go ahead. So the basic idea is there's this protein kinase called PKR, and it's part of your immune system, and it's in every cell you have. And what happens is when one of your cells gets infected with a virus, the genome of a virus is so small that in order for the genome to have all the genes it does, some of the genes have to overlap. And so a cell infected with viruses will definitely have double-stranded RNA. But one of your normal cells will never ever see double-stranded RNA. So the question they're asking is if I put any double-stranded RNA in the cell, am I going to get twitching? Right? Is it just part of the panic or immune system response? If I put double-stranded RNA for hexokinase instead of UNC22, am I going to get an UNC22 phenotype? Right? Am I going to get the twitching because it's an immune system response? And what they found is no. I want you guys to go around and I'll draw the next figure here. Okay, so the next thing is the tail end of the paragraph. This is super cool. So let's say, sorry, meant to leave it there. Let's say this is the UNC22 messenger RNA. Okay? And we make antisense, that's this. And we get all our UNC22. It's not the immune system panic response that's causing twitching. So this was amazing. I love what they did here. Is they did recombinant DNA technology, and they created a sequence, and they stuck that on the end of the messenger RNA, like this. Right? And then the antisense that they designed hybridizes here. into this non-UNC stuff. And the question is, if I make this the double-stranded RNA, do I shut down expression of UNC? And the answer is no. So even if it's attached, so it's sequence specific. What if they put some other gene on here, they'd be shutting down the other gene. And UNC22 would not be shut down. That's what they found. All right. Thank you. What are we on? Group 5? We're doing great on time. Yeah, put my paper down. I'll number here.
Huh? That arrow on the bottom.
Yeah, arrow down.
Up. Okay. There we go. Thanks. Well, I'm Eric.
I'm Molly.
I'm Zach.
We did the examination of paragraph 6 of the 1998 microRNA paper. So here's some basics for a paragraph. So they talked a lot about the null mutant phenotype, and that just means that there's a mutation in the gene that leads to the gene not being transcribed or translated. So it's just a really easy way for them to see if the gene got transcribed. And then, so in their experiment, the objective was to test the efficiency of the sense and antisense single-stranded RNA, and also the double-stranded RNA. And they also tested specific regions on there, so like specific genes on the antisense single-stranded
Yeah, and that's playing off of the last paragraph where we're seeing if we put something here, does it inhibit the rest of the gene as well? So this is a figure of the 4 genes that were studied, UNC22, and then they also added 3 more that also have discrete, well-known phenotypes. So they could tell if the wild phenotype versus the null mutant phenotype where it didn't express the gene. And each of these little strips is a section that they created sense, antisense, or double-stranded RNA for, and that's the area it would be targeting and seeing if that would shut down gene activity or not. And like I said, each of these extra genes that they were testing for had well-known phenotypes, and it was obvious if it was actually expressed in the progeny or not.
So for the experiment, they took the RNA sequence of the specific regions that they were testing, and they did the sense single-stranded RNA, the antisense single-stranded RNA, and then the double-stranded RNA. And so they stuck it in a nematode, and then it hatched some eggs, and then they waited till the eggs hatched, and they tested the phenotypes in that progeny.
And so basically, they observed that double-stranded RNA interfered fully, and they got the phenotypes that they expected. Like the FEM1 was needed it encoded for a protein that was needed in sperm production from hermaphrodites. So when the null mutant phenotype of that was no sperm production. And this was all to see the target specificity of double-stranded RNA, and they couldn't see any significant interference with single-stranded RNA, except for UNC54C, and the null phenotype for that was
Paralyzed.
Yeah, they were paralyzed, but in 54C they just had undeveloped embryo and larvae. And there's that. And
Any questions?
Okay. Okay. Can you go back to the UNC54 thing real quick? Yeah, down. Oh yeah, go up to that figure here. Keep up again.
The one with all the genes on it.
Yeah, with all the genes and all the bars
Actual
showing
figure.
run mutations.
Oh, okay.
This one right here. Okay, so what they found with the UNC54. UNC54 is another one of these muscles in control. You get a twitching phenotype. So all the UNCs are isolated. The assay was, does it have smooth gliding or does it have uncoordinated gliding, right? So in UNC54, one of the sense antisense, they make double-stranded RNA and they put that in cells, and basically the organisms died. They couldn't move at all. So what's going on with this? Some of the UNC54s they did twitching phenotype. There was one they did and the organisms were dead. It turns out, if you look where they did that, in here, and it's probably this one right here, one of these guys right here.
It was C.
Huh?
It was C.
It was C? This one right here? Okay. So it turns out the sequence found in this gene right here, the nucleotide sequence that codes for those amino acids, is also found, that nucleotide sequence is almost perfectly conserved in like 20 different genes. So that if you're knocking out the UNC54 gene with that sequence, you're not just going to knock out UNC54, you'll knock out every gene that has that sequence in it. You'll knock 20 genes out. And if you knock out all 20 of the genes involved in muscle control, the thing dies. So what it says is it can be very specific to a gene if your gene has a unique sequence. If your gene has a sequence common with many other genes, you could actually knock out a whole set of genes by designing your sequence antisense double-stranded RNA properly. Does that make sense? That's incredible. It's amazing.
Does this like work in humans as well?
Yes.
So like you you know, like you can like poison someone
Yes.
and just like kill them with like
Yes,
double-stranded-
absolutely.
Okay.
Homeland Security. Okay, what are we on? Was that 6? Yeah, 6 want to go? 6 want to go? Yeah, we're good. Go.
Always good to have a backup plan. What? I feel like 8. I don't know.
Introductions? Oh, there it is.
I think this is like the second slide.
There you go.
There we go.
Okay, I'm Phil. I'm Alex.
I'm Kevin.
So our paragraph's pretty generic. This kind of reinstates what we went over in the last few. So in this first sentence, it talks about the phenotypes seen in cells that the double-stranded RNA was injected had a very, very high rate of knocking out the gene. And so basically just that it confirms what we were saying earlier with there's a high correlation between double-stranded RNA injection and the gene not being present, or I guess expressed. And this
these genes that are knocked out are, I guess, in conjunction with the defective muscle cells that they were targeting, as described earlier, and other hypotheses we tested.
So this sentence basically says that they use fluorescent proteins, 2 different types, to show that the double-stranded RNA is the one that actually affects the body muscle proteins. And that's pretty much what this sentence talked about. And then as the double-stranded RNA was increased, like the amount of RNA was increased, the amount of cells that actually fluoresced decreased, which was a little interesting. So.
And we reference figure 2, which helps what explains here.
So, yeah, figure 2 basically shows there's things that are glowing, and then there's no double-stranded RNA here, and then after they use double-stranded RNA, there's no more glowing protein, so they inhibited the production of those proteins because they aren't there, and you can see that. Nice visual evidence. And the final sentence, basically just saying what the picture showed, that after they injected double-stranded RNA that targeted the glowing proteins, there was no more glowing.
Yeah, and then the tag they use was just GFP.
Yeah, those are making fancy. It's just a generic tag that makes things glow to make it easy to see.
Any questions?
Okay. All right. Thank you. So actually, can you put that figure back up?
Yes.
I'm sure there's more information.
That one? Yeah.
Here, but
Okay. So I'm trying to remember this here.
it seemed like your key point was
Yep, there was
glowing
something.
and then not glowing.
So,
No, sorry about that.
You were just messing
I
up.
signed out and didn't like it, so I do go. Okay, here, finally. Anyways, that's the one you wanted.
Yeah, that's it.
So, I think what they did here is, okay, so these cells, so in here we have a thing that codes for green fluorescent protein. And when you have an organism that expresses green fluorescent protein, all the cells line up like this. It turns out this organism, they've stuck a gene in it, and then they stuck another gene in it. So they put green fluorescent protein into all the cells here. It's in the genome, so all the cells express it from the chromosome. And they put GFP in there a second time. And this one, they have the GFP code fused to another code. So it's one single protein, and this is called LAC-Z. And I think this one goes into the nucleus, and this one stays in the cytoplasm. This is off the top of my head from a year ago, I'm remembering this. So I think what they did is they designed an antisense that hybridizes right here. So both of these guys ought to be knocked out. And they do that, and you get this. And then they designed an antisense, so basically you get a double-stranded RNA in which it hybridizes here. And that's the double-stranded RNA. In this context, you should knock this gene out, but the cells should still be green because of this. And you can see there's some residual green here in this situation. So they showed, if you knock this out, it's not that you're going to knock out this plus every other GFP. You're knocking out this RNA specifically. And it's not like reading down and saying, oh, well, then anything else with that sequence. Does that make sense?
I think that's where it was. Yeah. It's really tricky stuff.
And they didn't really mention any background.
Yeah, it's only in the figure,
Yeah.
right, yeah.
It's hard to get on that.
So, is that sequence like are you still including the sequence that tells it to localize in cytoplasm, and then after it has the sequence that tells it to localize in the nucleus, or is that?
I'm starting to run out of brain power here this hour. So you asked?
So that GFP sequence, it probably has some sequence that tells it you need to localize in the
Yes.
cytoplasm.
Yes.
That second portion there that you had fused, does it have both sequences that localize in both regions?
I think this one has a sequence that says go to the nucleus, and this one has a sequence that says go to the cytoplasm. Or vice versa, I don't remember. Okay. I don't think we have time for group 7. Who's group 7? You're up first next time. So we have 7. Who's 8? Group 8. Who's group 9? You're group 9? Or are you 10? 10. So group 9 is Orlando and Katie. And they're going to apparently send me a video on YouTube, and we'll just watch that. And we don't have a group 11, do we? No, we do. Group 11. You guys have the amazing opportunity to present an entire paper. You guys are not going to believe what's in this paper. It's going to change your thought about a lot of things. All right, cool. Turn this off. Yeah.